Sir at Molecular Biology in Agriculture Division, Bhava Atomic Research Center, BRC, and so far major project is concerned, sir has completed almost 11 major research projects from sponsored by DBT, DST, GSBTM, and other agencies. Sir has many publications in his credit. He has published more than 61 papers, 53 from International Journal of having good repute and impact factor. Sir has guided many PhD students. 21 student has completed and 2 is registered. And so far, this type of crash workshop lecture is concerned, sir is associated with this since 15 years. And whenever we approach for such type of program, in spite of his busy schedule, he never said no. So really, we all are fortunate to have Naresh sir as a resource person in this particular crash workshop. So these are the keywords, and without wasting much time, I humbly request Naresh sir to please enlighten us on lectures biochemistry and metabolic engineering, basic concept and design of metabolism. Sir, please. Sir, over to you, sir, please.
Okay, good morning to all of you. It's very nice to give a talk on to the young students. And I'm going to show you my presentation. Can you see my slide?
Yes, sir.
Okay. So I'm going to talk today on basic concepts and design of metabolism. And metabolism is synonymous with life. Metabolism is a net effect of thousands of biochemical processes coordinated in a particular way. And this is what we call a macroscopic phenomenon. We know the individual biochemical processes are there. Metabolism is a net result of that. So the metabolism becomes a central thing for understanding all the life processes. And this metabolism comes in the two components. One is the structural aspect. So whatever the molecules and where these things are operating. So that depends upon the size and how many compartments are there. So we'll talk about that little bit more detail. And then these there happens in a it's a dynamism, it's a function. It happens in a particular location and the space in the whatever the cellular concept, and time. Time is also very, very important. So the metabolism is a net result of all this process. We'll try to look at the different aspects, how this metabolism operates in the living systems. And all of you must be known about this, that metabolism has two major components, classify all the processes into two. One is a catabolic process where the complex or whatever the nutrients are there, depends on the organism. These are converted into energy, and this energy is used by the organism for various processes. And it also produces some of the components which are required to make the life itself. These are what we call the anabolic process. So all these anabolic processes are making the cellular constant, whereas catabolic processes are producing energy. When we talk about energy, all the time we know that ATP, adenosine triphosphate, is the major one. And the anabolic process, it is the NADPH. That is how entire metabolism can be visualized in a overall perspective. What we need to know is that the metabolism is more governed by catabolic process than anabolic process. Generally, it depends on the context, more than 50%. sometimes even 90% of the energy, whatever the metabolism, is only geared for energy purposes, and it is, you can see there are non-dividing cells also utilize a lot of energy. The brain is a very simple example. But of course, it doesn't mean that there is no anabolism. There are a lot of protein synthesis and the thing going on. But most of the energy is going in these non-dividing cells for the other functions than the anabolic process. So that is the overall perspective. And all of you are familiar with why ATP is the most important molecule or the energy currency of the cell. This is because of the phosphodiester bonds, which have a high energy. It can activate various groups in the living system, that is the hydroxyl groups of the amino acids and carbohydrate moieties and so on and so forth, and NH2 moieties of amino acids and other things. So this makes the, if you remember some of the organic chemical reactions, it makes the all the reactions activated form so that they can happen at the room temperature. So the beauty of the energy metabolism is there are some processes which are happening, majority of the processes are happening in the aqueous environment, and some of the things are happening in the lipid and non-aqueous environments. But ATP is the major molecule which is involved in the energy transfer. Although we have all the triphosphates are present, the ATP is the most important molecule. So we really don't understand why ATP was selected as the high energy molecule compared to all other 4 nucleotides. This type of question we have there for in living system. Why only 4 bases are taken, 20 amino acids are taken, typical sugars are taken. These are the boundaries of chemistry and biology, which remain the major problems. Why? But interestingly, although ATP is the most high energy molecule used in the cell, this is not the one which has the highest energy. There are so many other molecules are there with equivalent or even high energy state, but ATP is chosen for this. That is a mystery for us, the biochemical mystery. Another very important molecule is majority of the things we have some sort of a oxidation reduction reactions inside the cell. These are in the catabolism, you are generating oxidizing complex or reduced molecules, organic molecules, into more oxidized state, and that energy is used to make to the ATP and so on and so forth. And so you need an oxidation process, some sort of a molecule which can take those electrons. That is mediated by NAD. So NAD and NADH, the oxidized form is NAD and the reduced form is NADH. And this is the thing which is responsible for most of the catabolic regulation. And we will also see that this ratio is the thing which is also going to govern how much ATP is going to generate in majority of the process. And similarly, we have another related molecule of NAD, that's NADP. NADP and NADPH. The anabolism is a converse of catabolism, where you oxidize molecules, you convert into more reduced and molecules. For that, again, you need electrons. But interestingly, living systems have chosen again another molecule, NADP, instead of NAD, and that is responsible for the anabolism. And in addition to NAD, there are other cofactors which are involved in the oxidation reduction reactions. These are FAD and FMN, but the NAD is much more important than other cofactors which are involved in the oxidation reduction. And compared to NAD and NADP, as we have already seen, that catabolism is much more dominant. It is reflected also in terms of NAD levels. NAD levels are 10 times more than NADP in the systems. So this is how the metabolism is mediating its functions through using these cofactors. And ATP generation, there are two ways you can generate ATP. One is the normal, straightforward reactions where the oxidation of the substrate directly generates in ATP molecule. These are the process which are mainly involved in the glycolytic steps. And we need to understand in the metabolism, although there are so many processes utilize various ATP generation, NADH generation, and so on and so forth, whose contribution is more. So this is mainly depends upon what is the input thing, what nutrients, what the living systems are taking. So most of our central molecule is glucose organism. And of course, bacterial systems have much more complexity. We will briefly introduce that. But majority of the cases, it is dependent upon directly or indirectly on glucose. So glucose getting converted into pyruvate becomes a very important step in the generation of ATP. This is what we call substrate level phosphorylation. Then we have other thing where pyruvate is getting converted into carbon dioxide. That is through your TCA cycle and electron transport. And this is what we call oxidative phosphorylation. So ATP generation by two different processes are there. Energetically, one glucose molecule generates a net of two ATPs in the glycolytic process, and in the oxidative phosphorylation, it is a 36 ATP molecule. So very high energy of ATP generated in TCA cycle. So generally, there is a sort of a misconception that wherever you require high amount of ATP, mitochondria and oxidative phosphorylation should play very, very important role. But it turns out in the animals and so on and so forth, actually, when you require a very high amount of ATP in a short time, living systems employ glycolysis. And one of the examples is the cancer cells, which is utilized, they require a lot of ATP, and they use glycolytic process. Only thing here is they use a lot of glucose for their consumption. So this is what is known as aerobic glycolysis. So this also known as Warburg effect, and this is almost 100-year old phenomena. So don't be under the impression that glycolysis is not very energy ATP generating component. But majority of the cases, the oxidative phosphorylation is the major source for generating of ATP. And in the living system, we have bacterial systems are the single one. So everything is made in that. Whereas in the case of animals and plants, ATP is generated predominantly in mitochondria, and in plants it is chloroplast. But in plants depends on the cell types and conditions, whether chloroplast or mitochondria or both are involved in the generation of ATP. So how this ATPs get generated? By oxidation of pyruvate to acetyl-CoA. This was the fascinating problem which was there for a long time, and Peter Mitchell has demonstrated us that is not like a simple phenomena. It is the process of the oxidation reduction. He is directly not generating. It is only generating some sort of a gradient, proton gradient, and this proton gradient can be used for ATP generation. So it is very important for most of the us to realize that the electron transport function is not to generate ATP, it is to generate proton-motive force. Um, and some of the time, this proton-motive force, how it is generating ATP is mediated by this enzyme, what is called the ATPase. And this is a very fantastic enzyme. You are going to learn more details of how it is, or you might have already learned how the proton-motive force is converted through this beautiful enzyme. You can see that it's very complicated enzyme. There is a membrane components and structure and how it functions. It's a anyway, we'll not go into the details, but this enzyme evolution is become a very, very important for the organism to make ATP from proton-motive force. So in addition to this, we already talked about NADPH, which is required for. One more thing. In the glycolysis, as I said, substrate level phosphorylation is there. You also generate NADH. That NADH and whatever is generated in the TCA cycle, all those reduced cofactors are being used through electron transport to generate proton-motive force, and that is what is generating there. Some amount of proton-motive force is converted into ATPase. In addition to this, we also have this NADPH generation, which is required for anabolic process. So there is a process from glucose 6-phosphate going into ribulose 5-phosphate. In that, there is two steps where glucose 6-phosphate gets converted to 6-phosphogluconate, you generate one NADPH, and 6-phosphogluconate getting generated to ribulose 5-phosphate, it generates another NADPH. So these NADPH is the central thing for the anabolic processes. So remember that pentose pathway is some amount of glycolysis is diverted towards pentose pathway when you require anabolic processes. So in the cancer cells or something, or rapidly growing cells, the pentose pathway becomes much more dominant. So the overview of metabolism is the complex molecules are converted into simple molecules by catabolism, and simple molecules are generating again cellular constituent utilizing NADPH. And in the catabolism, you are generating proton-motive force and ATP, and simultaneously you are generating whatever is involved in NADH and NADPH are the intermediates which are responsible for the process. So, you can see the molecule in terms of complexity of the molecules. It is showing the glucose. Glucose by glycolysis, it's converted into smaller molecules, simple molecules, and the simple molecules makes many precellular constants, simple monomers, and these monomers convert into macromolecules at this. So this is entire process is your anabolism. So anabolism is much more complex, but in terms of amount of what is required is the catabolism is the amount-wise catabolism is much more dominant. Anabolism is less, but still this what one of the things drives energy to you make this cellular complex. So in addition to that, cell is doing many other functions. For that you require energy. First, the nutrient uptake that itself requires energy. In the case of simple bacterial system, it doesn't utilize ATP. Many of the transport functions in the bacterial systems is directly utilizes proton-motive force. So that is why it depends upon the organism in the because the bacterial things, many of them have only single membrane, that is from the outside environment. So the generating of the gradient of the membrane potential and proton-motive force becomes dominant role, and that can be easily utilized for various transport process without spending ATP. That's what happens in the bacterial systems. So now we will look at these phenomena in this process happening in a cellular context. So we are talking about a bacteria. This is the size, one plasma membrane. All the processes are taking place in this, and this is size is about 1 micron. If you are looking for other cells, whether it is a fungal cell or animal cell or a plant cell, they are much bigger, at least 10 to 50 times bigger than this. And if you take some cells, neuronal cells, they're extremely big ones. So, and what we need to also understand is as the magnitudes of the molecules and the cells increase, the problems become more complicated. So that every fold increase, because the volumes are going to increase. So you remember whatever metabolites and everything, the concentrations are going to change in orders 10 to the power of 3. That is going to make a very, very important contribution. This is the concept all of you should realize, because the amount of metabolism has to cater for bigger systems is going to be much more. And what we know is the smallest bacterial system is Mycoplasma, which is about 300 nanometers in size, and its volume is something like it has 10 to the power of minus 14 liters. See this ribosome size is 20 nanometer. So the bacterial cells have very, very small with respect to this, whatever the molecular constraints which are responsible for making that. So if you look at all kinds of organisms, it is glucose, we have given an example, but there are different ways you can generate energy. You can use various chemical molecules. Basically, it is chemical energy is converted into the living systems, what is required for survival. Both electrons as well, whatever we talked about, generating NAD, ATP, proton-motive force, and all those things, whatever way you can generate, that is necessary for survival. So what we see is majority of the metabolism diversity, whatever is there, fungal animals and all the things are predominantly using the chemoheterotrophs, that is the using organic molecules to generate all this ATP and NAD, whatever is required. And we have, on the other hand, there are organisms which can also use light as energy source and convert it. So this is the diversity of living system. And this diversity is extremely high. Metabolic diversity is extremely high in the lower systems. Bacteria, archaea have more complex metabolism. Fungal systems have little bit less, and as it goes towards the higher systems, plants and animals, they have limited, but we'll see what is the outcome of that. So, so what we are looking for metabolism, when we're talking about all the living systems, all of them, interestingly, they make same molecules. DNA, RNA, proteins, all of them are same. So there is some sort of a diversity, unity is there in the diversity. Irrespective of the living systems, all of them have to converge to make the same component. That is the beauty of the system. That is what we are looking for: metabolism, unity in diversity. Although each organism has diverse metabolism, we are looking for the common principles. So this is a typical bacterial cell of 1 micron in size. And interestingly, this organism has evolved on Earth something like 3.7 to 3.5 billion years ago. And our Earth is something like 4.8 billion years. And few 100 years in the early, the temperatures was very high. So maybe 0.5 billion years was went on the Earth to cool. So probably these bacterial systems, what are the first living systems, have evolved in a span of something like 1 billion years. This is the most one of the complicated problems, what we call origin of life. Anyway, that if you look at this any bacterial system, it has something like 15,000 ribosomes and something like 1,000 proteins and enzymes, which are component, and they are doing some 1,000 metabolites and cofactors. And it has one single DNA molecule which is orchestrating all these processes. That's the molecular perspective of a simple bacterial cell. And if you look at the inside the cell, because already said the initially we thought like a C type of structure where the proteins are floating like boats in the sea and everything. That picture is not true. It's a very, very complex, dense system with so many molecules are there. So this is the current perspective of even bacterial systems. Okay, so what do we know about the metabolism in bacterial system now? We know most of the biochemical pathways. And how do we know that we know them? One is that we are able to modify the existing pathways and design whatever we wanted using this metabolic engineering, pathway engineering, and so on and so forth. And we are also able to create new pathways. And the next level, what people are doing, that because our knowledge is so much, we are designing the microorganisms to do specific purposes. So using the microbial system to produce various products, metabolites, and everything of commercial benefit. Now you reduce the structures, microbes are there. Many of the things which may not be required for survival and their basic properties in a defined environment. So the making the microbes very, very efficient. So this is the one of the hot topics which is going on now. This is called microbial chassis. And then we have already seen that almost 10, 15 years back, we already created some minimal cell synthetic life. So we know minimum biochemical pathways are responsible for a living system. So what are the challenges remaining? Which pathways and metabolism operate in their natural environment? If you realize more, you might be learning something about E. coli, metabolic pathways, and so on and so forth. Glucose becomes the central pathway. We know the preference for glucose and so on and so forth. But in the real system intestine, is really glucose important for the survival for its own system? That type of question we still don't have an answer. So that's a that type of thing. When how actually these bacteria are using, what type of metabolism operates in the living system. Second, variations of this metabolism in species and its significance. That means if you look at the E. coli and if you isolate, let's say, from the different regions of the world, you will have so much variation in their metabolism. So then how do we define that? That is another species identity and metabolic uh, correlation with the species identity and so on and so forth. That also we don't know. Next, the most famous problem, which is the unculturable microorganism. All of you may be familiar, but we can only culture less than 2% of the um, bacterial systems which are there in the natural environment. And some of the things now, a little bit improvement is there to understand the unculturable microorganisms, but this becomes a very challenging thing. So now we move on to next step, the eukaryotes. The eukaryotes have come on Earth something like 2.7 billion years. And I said 3.5 to 3.7 billion years prokaryotes have come. And it took about, again, 1 billion years to generate the animal eukaryotic systems. So almost in terms of complexity and of the problem, origin of life and the evolution of the prokaryotic world into eukaryote are equally or much more complicated. So, and here we can see that there are two types of systems: animal systems and plant systems. Plant systems have an organelle, which is chloroplast, and mitochondria, whereas the animal cell has only mitochondria, it doesn't have chloroplast. This again, how these things have come, it has come by the prokaryotes forming to the first eukaryotic cell. People believe it is again some sort of a combination of archaea and bacteria generating a nucleus containing eukaryote, and then it is engulfed other systems to bacteria to generate this mitochondria and chloroplast. If you compare the precursor of bacteria and these organelles, you realize that these organelles have become much more sophisticated for what their special activities. That is, mitochondria, you have a lot of invaginations and everything, cristae, which makes the mitochondria only for making ATP generation and electron transport, whereas bacteria is not true. There's only sometimes the bacteria generates ATP, but in all other cases, it's doing many other processes. Same way, cyanobacteria using the light source, it is generating energy. Now, but in the chloroplast, the entire functions have become there. So, if you now look at the, and we already said the eukaryotic cell is much bigger than the bacterial system. If you look at this mitochondria, you can see that mitochondria, each bacterial cell, if you assume this is a bacterial cell, there's so many bacterial cells are present inside the eukaryotic cell. So that is the complexity. Imagine so many bacteria being together, working in a coordinated fashion, and giving it a much more complex system. And you can see that the whatever the complexity is present in bacteria is a much more complex. It is a much higher organism, and cell biology is going. You are going to look at how these things are being well organized. In terms of metabolism, because of this, what you also realize, there are a lot of membranes are there. Each one is separate. So the compartmentation has happened within this, and the entire metabolism, what was happening in a single compartment in the bacterial cell or prokaryotes, it got divided into regions. You can see there is a cytoplasm, has many of the process. Mitochondria, we already said, is involved in the generation of energy and many other process. Lysozyme type of thing, which is in the degradation of molecules, macromolecules and carbohydrate, nucleic acid, proteins, and all the things. And the endoplasmic reticulum is responsible for generating biosynthetic proteins and everything. You need to go outside. along with the Golgi, and so on and so forth. Nucleus, of course, is involved in the complete DNA and RNA biosynthetic pathway. So now, because of the big size, each one has to become more sophisticated, and the compartmentation has divided that. So these specialized functions also, each one requires energy. Whatever we talked about, ATP, NAD, NADPH, all of them are required. So this is the what is the benefit of this? Because metabolism has become much more efficient. Each one is able to segregation becomes much more efficient systems. The but once you segregate this, then you are going to generate a new problems. What is that? The distribution of coordinating all the whatever things are there into different cellular compartments. Here, if you are saying that ATP is generated in the mitochondria, NADPH is getting generated in the cytoplasm, but how the ATP and NADP goes into different cellular compartments. Everywhere you require some sort of a requirements of these metabolites and everything. So distribution in the eukaryotic system becomes very important. And the energy requirements for all the process which are taken similar to that in prokaryote, the eukaryotes require much more because the complexity of the systems are more. That is structural proteins, transporters, and so on and so forth. So what we have already told you that glycolysis is a substrate level phosphorylation, although it is inefficient in terms of number of ATPs can generated from one glucose molecule, but it is very fast. Where the mitochondrial oxidative phosphorylation is slow process, but it generates a lot. So the organism, the cell eukaryotic cell, it coordinates these biochemical process fast and slow as depends upon the requirement. Other important things are the membranes, how membranes themselves are formed. These membranes are formed, little fatty acid synthesis, everything and the complex thing is taking place in the endoplasmic reticulum, and these membrane components have to be distributed. So we don't know too much about this. But out of all this process, you require some sort of a, we ignore the ionic interactions when the molecules become big, and that also charged molecules. So, and if you involve this lipid molecule transfers, we know within the cells it happens by some sort of a pinching of vesicles and fusion of vesicles, and that's a dynamic process. The fusion of vesicles, because these are phospholipids, the negatively charge is there, you require some sort of a neutralizing thing through ionic interaction so that the fusions can take place. Calcium becomes very, very important in this context. So eukaryotic cells, the calcium, if you have high levels, there's a serious problem because the phosphate calcium can form, they're highly insoluble, they form complexes and it becomes a complication. So the eukaryotic cells has to coordinate the calcium levels in a particular environment, in a transiently, very small concentrations, suddenly produce that. That is the additional contribution has come to facilitate the communication, lipid transfers and everything between the cells. And one more aspect of this, because the glycolysis is in the cytoplasm, the pyruvate has to enter into the mitochondria, and then the NAD, NADH level ratios also compartmentalized. So every time the distribution of this metabolite is not optimal within the cell. So one example is I give you here. where electron transport you require NADH, giving electrons to through the proton-motive force generation. But if you don't have enough amount of oxygen, or the in-between components are not, the process is not very efficient, then there is a transiently the electrons remain in the electrons component, and oxygen is present all the time. The eukaryotic systems have, most of the things have been made such a way that oxygen is present around, and that is going to be very complicated problems, because oxygen is an unusual molecule with triplet at the ground state with two single electrons. So it immediately takes electrons from that and becomes the reactive oxygen species. There are many oxygen species are there. This generates a serious problems within the cells, and this reactive oxygen species can damage any component, amino acids, cell wall components, plasma membrane, lipids, everything. So this requires some sort of a controlling of whatever is generated is there to take care of that. You convert oxygen radicals, you give electrons and convert to ultimately water molecule, and that requires NADPH. So the NADPH requirement in the most of the higher eukaryotic systems is not only to make NAD, anabolic process, but it is also to take care of various reactive oxygen species controlled phenomena. This we have realized that the regulation of these oxygen radicals is plays very important role in the higher systems in terms of proliferation, differentiation, autophagy. We are going to talk about some of the things in later, when the immunity, aging, and so on and so forth. So oxygen radicals is a central thing. Regulation becomes there. So there are many antioxidant mechanisms also cell had to develop, and this happens in each and every compartment. So every compartment you need to have NADPH to, and whatever is the system. Glutathione is the molecule, a small peptide which is involved in the neutralization of this reactive oxygen species. So you need NADPH and glutathione in each and every cellular compartment. How is it there? We still do not know very clearly what is the way the endoplasmic reticulum and mitochondria are managing. We know a lot of things what happens in the cytoplasm. So in addition to that, there till now we talked about small things, but the macromolecules are very important. In the eukaryotic system, there are so many proteins, so many macromolecule numbers that become very important molecule. DNA itself is a big one, and it involves various modifications within the methylation, acetylation. These are involved in the maintenance of the structure and functional state of the genes and so on and so forth. Then another problem is in the protein synthesis, because of this compartmentation. In nucleus, you have transcription, the mRNA has to be processed, and it has to go to the cytoplasm, and then it has to be made into proteins and so on and so forth. This is a very slow process. But many of the time, biochemical reactions require very fast responses. So what these cells do, they keep these proteins and enzymes in a inactive mode, and then they convert into active state. So that is involved in a lot of protein modification. That requires a lot of energy. In addition to this, because macromolecules are there, and they stay very long, as we have said in some of the cases, like maybe few years to days, years, and so on and so forth. So the molecules have to be maintained. DNA. So the reactive oxygen species, whatever things are going on, there's some sort of a damage is going to take place. So they have to be repaired. Enzymes which are going to be protected for the degradation. So that is a process which is called acetylation, and lipids are going to peroxidation. So there are a lot of maintenance problems are there in the cell. So a lot of energy goes on in maintaining the macromolecules. So the metabolism has to gear to so so in the eukaryotic systems, the metabolism is geared more towards maintaining of these complex structures, and then coordinating the whatever the requirements are there within the different cellular compartments. So and if you look at the these eukaryotic cell, then each cell is a part of the multicellular organism. It's a complex system. So whatever have till majority of the information what we have come from the in vitro studies. But in vitro studies we realize may not correspond to in vivo, because the context is different. So many things are there. We realize that now the metabolism of cell happens, what happens in vivo, same cells in different location, context wise where they are, that is going to make a lot of difference. So that is one of the major problem we need to understand. Second, like what we said, simple biochemical pathways to metabolism. Now in the multicellular organism, the cell metabolism in coordinated fashion in a tissue and organs then becomes physiology. So that is the next level. Currently, the techniques have become powerful. So what I believe is that now we are progressing from in vitro cell metabolism to in vivo cell metabolism. Once we look at, and the techniques have become extremely powerful to monitor in vivo metabolism within the living organisms. So we are looking close to understanding the cellular metabolism in physiological context with anatomical and tissue organization. So this is a fantastic future is there. Probably the cell-based studies are going to be less significant, and we are going to go towards more realistic and case of understanding the metabolism. So the same process, so initially, now current perspective is almost every biological phenomena, people are looking at the basic metabolism point of view. So this we are able to reach that, and that is a current perspective. Now only thing is, a lot of things are looking at the human and perspective point of view. Hopefully in the coming years we also have more understanding the similar type of thing with the plant-based system. So that is the overview of the metabolism. Thank you, and best wishes for all of you. Now I am open for any sort of questions.
Dear students, if you want to ask any question, please ask.
Please ask your questions. I have requested you. Dear student, please ask questions if you have. Dear participant.
हेलो सर।
हाँ पूछिए
सर हिंदी में कैसे बोल सकता हूँ?
हाँ बोल सकते हैं
सर सीनियर सिटीजन में कैटाबॉलिज्म वीक क्यों हो जाता है?
Oh, simple. As I said, damaging is taking place. You have this, whatever the metabolism is generating various molecules, and you have a damage is going on all the time. So, repair process are also going on, but over the time, the damage takes over. Simple. Any system, it's like any other machine. Over the time, it goes bad. You keep maintenance. If you keep maintenance well, then it will last longer. Exactly the same thing goes on in our system also. If we are able to take care of this repair process in terms of this reactive oxygen species mediated process and so on and so forth, probably we can delay a little bit. Otherwise, it's a natural process.
Okay, sir.
Yes, dear student. Any doubt? Please ask. प्लीज डोंट हेजिटेट. आप हिंदी, इंग्लिश, गुजराती सर को पूछ सकते हो, कोई दिक्कत नहीं है.
सर चैट बॉक्स में है किसी का?
सर चैट बॉक्स में एक क्वेश्चन है.
Yes,
sir.
I saw that. That's it. We don't have an answer for that. Simple. Why GTP is used instead of ATP or other things? These are some of the challenges. Hopefully, sometime we will get the answers. Like same way I asked you ATP why this is used, and CTP and UTP is used mainly for carbohydrate activations and so on and so forth. So we don't have an answer for this.
Any more question by anyone? So, Amit sir.
Yes, sir.
Should we conclude?
Yes, sir. You please conclude, sir. Please.
Okay.
Yeah.
So, sir, thank you very much for such a informative and excellent lecture. So far, competitive point of view is concerned, and sir has already covered maximum and entire important concept of metabolic process, particularly plants, animals. Sir has also covered modifications of DNA sequences, other sequences that perform a specific metabolic action. And so far, TIFR, IIT, JAM or other competitive exam is concerned, you will find it very useful because 2, 3, sometimes 5 questions may ask from this particular area, and sir has covered it nicely. So, behalf of GSBTM and NV Science College, sir, we thank you. We pay our gratitude to you for sparing valuable time. Thank you very much, sir, and we seek your blessing as well as favor in future also. Thank you. My regards, sir. Thank you very much.
It's my pleasure.
Thank you. So, sir, can we give break for 10 minutes?
Yes, sir. We are giving them break for 10 minutes, and they will be joining the next link for the lecture 4 that they have already received.
I hope you listen, participant. Now you can take leave for 10 minutes and then you can join for another lectures. There, Dr. Ujjwal Trivedi sir will be there and he will be covered aspects of biomolecules. So now you can take leave for 10 minutes and then join before 10 a.m. Thank you. Thank you all. Thank you, participant.